You know, proteins are these incredible microscopic machines that literally build and run our bodies. But here's the crazy thing, they all start out as just a simple, floppy string of molecules. So, how in the world does that string manage to transform itself into a complex, working machine? Well, let's dive in and find out. And that's really the central puzzle right there. I mean, it's one of the most elegant processes in all of biology. How does a one-dimensional string of information, basically a line of code, know how to fold itself into a three-dimensional object that can actually do something? It's kind of mind-blowing.
Well, the secret is that it doesn't just happen all at once. It's a step-by-step journey, a process that unfolds through four distinct levels of structure. You start with that simple chain, and you end with a complex functional protein. So let's break it down level by level.
All right, first up, we've got the foundation of everything, the primary structure. This is ground zero. This is where it all begins. So, what is the primary structure? Honestly, the best way to think of it is like the protein's unique recipe, or maybe even its alphabet. It's simply the specific order in which these building blocks, we call them amino acids, are linked together in a long, long chain. And this exact sequence, this is the fundamental blueprint that's going to dictate every single fold and twist that comes later.
A perfect real-world example of this is the hormone insulin. Now, insulin's interesting because it's not just one chain, it's two. You've got an A chain with 21 amino acids and a B chain with 30. And let me tell you, the exact order of these 51 amino acids is absolutely critical. Just one little mistake in that primary sequence, and it can be the difference between a hormone that works perfectly and a completely useless string of molecules. Okay, so we've got our string of beads, our sequence is set. But it doesn't just stay a straight line for long. Now we get to the second level, where the first folds start to appear.
So at this stage, the chain actually starts interacting with itself. You get these weak hydrogen bonds forming along the chain's backbone, and they start pulling it into these really predictable repeating shapes, but just in certain local spots. And this stage really has two star players, two main patterns you're going to see over and over. First, there's the alpha helix, which, just like it sounds, coils up into a spring-like shape. And the second is the beta-pleated sheet, where different sections of the chain line up right next to each other, kind of like the folds in a paper fan. Now, digging into those beta sheets just a little bit more, there's a key distinction. The strands can line up parallel, meaning they all run in the same direction, or they can be anti-parallel, running in opposite directions. And it turns out that the anti-parallel setup allows for better, more optimal hydrogen bonding, which actually makes it the more stable of the two. And right here, you can see that difference really clearly. See how the anti-parallel chains form these nice, straight, strong hydrogen bonds? The parallel ones, on the other hand, are a little more angled. It might seem like a subtle detail, but believe me, it's absolutely crucial for the protein's final architecture. Okay, so we've got our coils and our sheets, our alpha helices and data sheets. The next step is to take all those little pieces and put them together into the final shape for a single chain. Welcome to level 3, the tertiary structure. This is the big one for a single chain. This is the level where the entire protein finally achieves its functional 3D form. All those secondary structures we just talked about, plus all the random bits connecting them, they all fold and crumple up on each other. And what's driving this? It's all about the interactions between the amino acid side chains, those unique parts that make each amino acid different from the next. A fantastic, classic example of this is a protein called myoglobin. This is the protein that's right there in your muscle cells, and its job is to grab onto oxygen and hold it in reserve. It's a beautiful, compact, globular structure, just one single chain folded perfectly to do its job. So, here's the crucial point to remember about myoglobin. It's a solo act, a lone worker. It's just one chain of 153 amino acids, and it's packed together so tightly that all its water-fearing parts are tucked away on the inside. And right in the middle, it's got this special heme group nestled in a pocket, all ready to bind to one single molecule of oxygen. Now, myoglobin is fantastic for its job, storing one oxygen molecule. But think about it. What about transporting lots of oxygen all around the entire body? For a job that big, a single worker isn't going to cut it. For that, you need a team. And that brings us to our fourth and final level, the quaternary structure. This is where multiple, already folded protein chains come together and assemble to form a kind of protein superstructure. To put it simply, This level is all about how two or more polypeptide chains, each one already folded into its own tertiary structure, fit together. We don't call them chains anymore at this point, we call them subunits. And when they combine, they create one massive, functional protein machine.
And the ultimate example here, the one everyone learns about, is hemoglobin. This is the protein that literally makes our blood red. And unlike myoglobin, which was just one single chain, hemoglobin is a complex made of 4 separate subunits, all working together in perfect harmony to transport oxygen from our lungs to every other part of our body.
So hemoglobin is this A team of 2 alpha chains and 2 beta chains. And just like myoglobin, each one of these 4 subunits cradles a heme group. So you do the math. One hemoglobin can carry 4 oxygen molecules. That's 4 times as many as myoglobin. But the real magic, the really cool part, is something called cooperative binding. What happens is, when one subunit grabs an oxygen, it actually changes its shape, and that change sends a signal to the other subunits, basically telling them to grab on too. It's like a form of molecular peer pressure that makes it unbelievably efficient at both picking up and dropping off oxygen exactly where it's needed most.
So, to wrap things up, what have we seen? From a simple sequence of letters to a complex, functioning team, every single level of folding is about one thing: achieving the perfect final shape. Because in the world of proteins, shape is everything. So let's do a quick recap. The primary structure is just the sequence, held together by strong peptide bonds. The secondary structure uses weaker hydrogen bonds to make those local folds. And then the tertiary and quaternary structures use a whole toolkit of different interactions to lock in that final 3D shape, whether it's for a single chain or a whole multi-chain complex. See? Each level builds logically, right on top of the one before. So why does any of this intricate folding matter so much? Well, because these incredibly specific shapes are what allow proteins to do, well, everything. They give us structure, like collagen. They transport vital molecules, like hemoglobin. They send signals as hormones. They defend us as antibodies. And they act as enzymes that run pretty much every chemical reaction in our bodies. They are the ultimate do-it-all molecules. When you think about it, this precise step-by-step folding is just a masterpiece of molecular engineering. It's absolutely essential for our health, for life itself, which leads us to a really crucial final question. We've seen how perfectly folded proteins build and run our world. But what happens when this intricate folding goes wrong? Well, that's a story for another time.